there. Welcome to the molecular biology series of lectures. This is the 16th lecture in this series, and in this lecture we will be discussing the different inhibitors of the translation process and the synthesis modifications which happen after the translation of the proteins, so that the proteins will go through this modification and generate fully functional forms which are folded and are able to do the dynamic or structural function. As now we are aware that translation is a multi-phase and multi-step process, it is very complex. Numerous compounds and drugs have been found to inhibit this process at any stage of the translation phases or in steps. Some of the important mechanisms can be either by preventing the initiation complex from forming and forming the initiation machinery which is able to generate the proteins, or by preventing the peptidyl activity of large subunit, or by preventing the translocation itself during the elongation process. Inhibitors of the translation is the puromycin, which is derived from the Streptomyces alboniger. It has a structural similarity to that of the amino acid tRNA, so it mimics its binding to the amino acid site of the whole initiation complex. So when it binds to the amino acid site, the peptidyl activity of the large subunit causes the peptide to be associated with the puromycin through its peptidyl activity, and it results in the formation of the peptidyl puromycin. This peptidyl puromycin is an inhibitor of the translation, as you can see in the accompanying picture. So it does not engage in the translocation at all. It dissociates from the ribosome shortly and taking the polypeptide with it. So it prematurely terminates the polypeptide and hence results in the loss of this polypeptide, which cannot function at all. Thereby it's one of the great usage in the pharmaceutical drug industry in working as the antibiotic. Important inhibitors of the translation are the tetracyclines and the chloramphenicol. Tetracycline acts as the blocker of the A site of the ribosome. It prevents the binding of the amino acid tRNA to the A site and hence prevents the translation process during the elongation phase of the translation. While as the chloramphenicol inhibits the bacterial protein synthesis by blocking peptidyl transfer. So chloramphenicol is utilized mostly as an antibiotic, and it cannot inhibit the protein synthesis in the eukaryotes, but only in bacterial. Important drug which has the capability of blocking the peptidyl transferase in the 80S eukaryotic ribosomes only is the cyclohexamide. You can see in the picture. And then there is a streptomycin, which we use also as the antibiotic because it causes the misreading of genetic code in bacteria at relatively low concentrations, and if it is taken at a high concentration, then it inhibits the initiation process as well. Finally, the diphtheria toxin, which catalyzes the ADP ribosylation of the specific histidine residue, which is called as diphthemide, within the eukaryotic elongation factor EF2, and hence inhibits it. So it is one of the toxins for the humans as well. And then there is a ricin, which is derived from the castorbrine. It inactivates the eukaryotic ribosomes at the 60S subunit, and this is caused actually by depurinating the adenosine residue within the 23S structural unit of the 60S subunit, and hence inhibition of translation in the eukaryotes. So moving to the next topic is the post-translational modifications. As we already know that the polypeptide, which is formed by the translation process, is not actually functional in nature. It is a pre-polypeptide which needs certain modifications, and those modifications are done post-translationally, sometimes also co-translationally also. So when these modifications are done, they attain the final functional form, which is folded properly to do its dynamic or structural function. Are these post-synthesis modifications or post-translational modifications which happen in the proteins and help them to attain their final fully mature structure, which is folded properly to do its function of dynamic nature or enzymatic nature or structural nature. One of the important is the deformylation. Deformylation happens for the initiating methionine, and it has been found that almost 50% of the eukaryotic and prokaryotic proteins do not contain the initiating methionine in itself in the functional form. So it is removed by methionine aminopeptidase. This is the first modifications which can happen in the almost 50% of the synthetic proteins which are synthesized. Then there is important another modification, which is an acetylation of the amino group, which is in the amino terminal. Usually 50 to 90% of the eukaryotic proteins are co-translationally modified by an acetyl transferase. This modification is usually done when a nascent polypeptide is being synthesized and is about 20 to 50 residues during the translation process in itself. So an acetylation happens as soon as it is around 20 to 50 residues in length and about in 50 to 90% of the eukaryotic proteins. This N-acetylation prevents them from the degradative action which can happen in the proteins as well. Then there is important another post-synthetic modification that is called as the removal of nuclear localization signal sequence, which happens for certain proteins which are to be packaged and moved from the synthetic machine. Post-synthesis modification is for those proteins which have a glycine at the C-terminal residue. And if the glycine is there, it is usually converted to an amide group. This modification is an enzymatic modification, happens in two different steps, which involves the utilization of ascorbic acid, copper, and oxygen as well. What happens for this glycine at the C-terminal residue is that it is first hydroxylated at alpha carbon. Then the glyoxylate moiety, which has just been created, is then removed by hydroxylation, and this removal generates the amide group from this glycine. And this is in the then functional form of the protein in its mature form. So many kinds of proteins which undergo a specific residue modifications for their functional state. Unless and until these residues are modified, they cannot function. So one of the residue modifications happens for the hydroxyl containing amino acid residues in the polypeptides, which are serine, threonine, and tyrosine. These contain hydroxyl group as the functional group, and these in most of the proteins they undergo phosphorylation, and phosphorylation is done by protein kinases, which utilizes ATP for the phosphorylation process, so that the final polypeptide which is formed contain these residues in a phosphorylated form. Another modification of the residue is of a glutamate residue where an extra carboxyl group is added. This is done in some proteins to produce gamma carboxyglutamic acid, and that this reaction, this modification is brought about by the carboxylases, which is vitamin K dependent and is active in the endoplasmic reticulum. So as soon as the proteins are synthesized in the eukaryotes, they go into the ER lumen, and then the carboxylate is added to the glutamate residues of these proteins, specifically for those which are involved in the cascade of the protein coagulation, blood coagulation proteins undergo these kinds of modifications necessarily. Another important post-synthesis modifications happens for the lysine and proline. This involves the hydroxylation of these two residues in those proteins which serve as the structural proteins, like for example collagen. In collagen, lysine and proline undergo hydroxylations to form 5-hydroxylysine or 3-hydroxy or 4-hydroxyproline. These are intertwined into each other and to give the more higher strength and higher structural stability to the structural proteins. And this hydroxylation involves alpha ketoglutarate, ascorbate, and oxygen as well for the synthesis of the hydroxylated form of lysine and proline. Another modifications happens for the glycoproteins. They are those polypeptides in which the carbohydrate moiety is attached to them. to form glycoproteins. Usually, the carbohydrate can be attached to two different moieties, either to asparagine or to either hydroxyl containing serine or threonine. If it is attached to the asparagine residue through N bond, they are called as N-linked oligosaccharides. And if it is bound to the serine or threonine through oxygen, then they are called as O-linked oligosaccharides. Another important post-synthetic modifications for the proteins, specifically for oligomeric proteins or sometimes also multimeric proteins, is that the creation of a bond between the two cysteine residues. These two cysteine residues result in the formation of a specialized bond. This specialized bond is called as the disulfide bond. It can be both interchain and intrachain, and it is done with the help of specific proteins called as PDIs, protein disulfide isomerases, which utilize glutathione as an oxidizer to create the bond between the two cysteine residues and to link them together. And this also gives the stability to the structural protein specifically. And finally, we know that there are some kinds of proteins which are synthesized as inactive form. Those inactive forms are called as zymogens. They are converted to a functional protein, which can be functional structurally or functional dynamically as hormone or as enzyme, by proteolytic cleavage. Some sequences get cleaved from the polypeptide so that it will be shortened, and then after it is shortening, it is functional in its shortened form. So this video completes the essentials of molecular biology in all their aspects, covering replication, transcription, and translation. So today, which is celebrated as the DNA Day, we completed the essentials of molecular biology. In the next lecture, I will be uploading something about the nucleic acids, and then we will see where we go further. If you like the contents of the video, you can order the book Molecular Biology and Biotechniques from Amazon. It's available both in Kindle and paperback format. And do subscribe to my channel for further videos which I will upload in due time.